This episode of the Ask a Neurologist podcast is brought to you by Brainvincible. Brainvincible is a company dedicated to promote brain health, and you can check out the products and services of Brainvincible at our website. You will find the link to the website in the description. So one of the products that Brainvincible has is this omega-3 fish oil. It's a highly concentrated fish oil with purified omega-3. It's free of heavy metals. I take this myself every day. So if you're interested in doing something for your brain health, in improving your cognitive performance, you might want to check it out. So my name is Rabea Akram. I'm a medical doctor. I am a co-founder of Brainvincible and also the founder of the Ask a Neurologist podcast. It's a podcast where I interview other medical doctors, healthcare experts, and research scientists who specialize in the field of in different topics about healthcare and brain and other health related topics. And today's guest is Dr. Adriano Maida. He is a senior research scientist at the Helmholz Center in Munich and at the Diabetes Center at the Institute. He has a very impressive research career with a specialization in the field of diabetes research. He has had numerous scientific publications in renowned scientific journals such as the Journal of Clinical Investigation and Gastroenterology. He has had years of experience in research in this field, and previously he was working at the Cancer Research Institute in Heidelberg, and before that at the University of Toronto, where he did his Ph.D. So I'm very excited to have this interview with him today. This is going to be a really fun interview where we get into the really details of science behind diabetes and the newest stuff that is happening in scientific research in that field. So if you are interested in metabolic health, in diabetes, in how different types of nutrients affect your metabolic health, so there is no better person to explain all that to you than him. And so I'm very excited. So let's get started. Adriano, welcome to Ask a Neurologist.
Thank you for having me. I'm happy to be here.
You're welcome. So you have so much knowledge about diabetes, and we want to know all about that. Maybe we start with the very basics. Tell us about where your interest in diabetes research came from, a little bit about you, where your passion lies, and how you decided to become a researcher.
So my career path started during my bachelor studies, so I was always interested in science. And after finishing my, close to the end of my Bachelor of Science degree, I had to choose a project to conduct with someone at the university at the time in Toronto. And I chose to do a project that was led by someone who was working on diabetes. It was a bit of a coincidence, in fact. So I was always interested in metabolism and how metabolism controls blood glucose, which is a major factor in diabetes. And then I started to do work with someone in Toronto named Gary Sweeney, and this was kind of the kickstart to the rest of my career, which was basically research to investigate how diabetes, what causes diabetes to occur, what are the mechanisms behind it. So from there, so I conducted some experiments and studies at York University to finish my bachelor's degree. And afterwards, I went to continue such research at the University of Toronto for my Ph.D. And then I became super interested in not only the mechanisms behind diabetes, but also ways to treat it. And so my Ph.D. years were about understanding different novel ways that we can reduce blood sugar in people with diabetes. And then that perpetuated my research into my postdoctoral years, which were actually conducted in Europe. And ever since I've been conducting work on understanding diabetes and doing finding new ways to treat it. So.
That's really amazing. Maybe in the beginning you can explain in a simple way for us to understand what the mechanisms that lie behind diabetes and the different types of diabetes.
So diabetes is generally a problem related to elevated blood sugar. And it's broken down into two types, so type 1 diabetes and type 2. And both of those are centered around the action of a hormone called insulin. And insulin is actually important for our bodies because when we eat, normally insulin levels go up, and this helps to reduce our blood sugar. In the case of type 1 diabetes, people are not able to produce enough insulin. And this is the type of diabetes which is more frequent in young adults and in children. And so patients with type 1 diabetes often have to inject with insulin several times a day in order to keep their blood glucose at a low level or at a safe level. In the case of type 2 diabetes, which is more frequent in overweight and obesity, as well as in the elderly, again, the issue is related to insulin, but these people often produce quite a lot of insulin, but the problem, the mechanism that relates to diabetes is actually that the insulin cannot function properly. So although insulin is quite high in these individuals, the blood glucose is not being lowered effectively. So in these individuals, in some cases are also treated with insulin as well, but other therapies are available, such as oral medications that help to improve the action of insulin. So in general, insulin is a hormone that's produced by our pancreas. And as I mentioned, in the case of type 1 diabetes, these cells in the pancreas, which are called beta cells, are actually, they're depleted. So there's not so many of them, and that's what leads to the lowering of insulin. And so in the case of type 2 diabetes, there's also a similar a problem that occurs in later stages, that the beta cells are either not functioning normally or are fewer in number relative to the needs of the body. And so strategies to improve the function and the number of beta cells in the body in both type 1 and type 2 diabetes are very, very relevant.
Okay, so from what I understand, in type 2 diabetes, in type 1 diabetes, there are less beta cells, and in type 2 diabetes, there are enough beta cells, but can you explain that part again?
So in type 1, the main origin of the disease, I would say, is an autoimmune problem. So the beta cells of individuals with type 1 diabetes are attacked by their own body. And so some individuals have beta cells remaining, but it's very, very few, and it's obviously not enough. So then they have to supplement their bodies with exogenous insulins by injection. And in the case of type 2, the issue is often related to insulin resistance. So although individuals have very high levels of insulin circulating, due to problems related to obesity or overweight or age, the ability of insulin to act in these cases actually is less effective. So in this case, the person has relatively not enough beta cells present to be able to meet the needs of the body. So that's why in some cases of type 2 diabetes, often in later stages, people are treating themselves with insulin as well, because it can work. It's just that the amount that they have circulating is not enough.
So your PhD research has been also related to these cells in the pancreas, the beta cells, which produce hormones called like gut-derived peptide hormones, and how these hormones play a part in sort of like diabetes and the development of diabetes. Can you tell us a bit more about that?
So my PhD work was focused on peptide hormones, which are hormones that circulate in the body, like insulin. But these are actually not coming from the pancreas, but from the intestine. So when we're sleeping or fasting, these hormones are low. And what triggers their release from the intestine is actually nutrient intake. So whether it be glucose or fat, this triggers the release of these hormones into the circulation, and they have a variety of functions. One of these hormones is called GLP-1 as a short name, and this particular one can, when it's circulated, it triggers a signal in the brain to reduce your food intake. So it's one of these feedback mechanisms that the body has, so that you slow down eating. And one of the other functions of this hormone is to act actually in the pancreas on the beta cells to promote insulin release. So together, with its actions also elsewhere, this actually causes a glucose lowering effect. And there are other gut peptides in this family which act in a similar manner to regulate not only food intake, but also the release of insulin on the from the beta cells. And as well, so this promotes glucose control, and as well, they have influences on the way that the beta cells perform. So not just being able to secrete insulin, but also on the beta cell number, on their long-term survival rate. So there are a number of benefits of these hormones, both in a physiological sense for the body to control glucose in general. And that's why I would say that since my PhD years, around that time, a lot of new drug molecules have come on the market which are related to the action of these hormones, which in general are called incretin hormones.
Okay, so are there currently therapeutic drugs which are working in this pathway through the gut-derived peptide hormones?
So on the market now there are several available. So one class is called DPP-4 inhibitors, which target an enzyme which in circulation actually degrades these hormones. So when you take these drugs for diabetes, the patients experience higher levels higher levels of these active hormones in the circulation. So that, for example, the GLP-1 that I mentioned is elevated to a higher level, so that during food intake, patients can achieve higher insulin levels, a better glucose control, and normally also it can also affect their food intake behavior. So this is one class of drugs which targets, I would say, both incretin hormones in this family. And then there's another class which is related, which are injectable incretins, which is mostly related to GLP-1. And nowadays there are others on the market which are related to others. But these are hormones which patients must inject. And with these injectable hormones you can achieve more effects on not just with glucose control and insulin release, but also effects on body weight loss and on food intake. So together, because type 2 diabetes, I would say, is also related in a strong way to body weight and obesity, they have pretty profound effects on reducing blood sugar.
Okay, so from what I understand, these peptide hormones are very important for also the beta cells in the pancreas, for them to sustain, right? And also for the entire regulation of glucose metabolism in the body. So are these hormones in any way affected by external factors such as diet, stress, or exercise? Is there any direct connection?
So in terms of diet, I would say they're strongly related in an acute sense by food intake, as I mentioned. So as soon as you ingest a meal, their levels go up in healthy individuals. They're influenced by diabetes as well. So in patients with diabetes, for example, they have lower levels of these hormones circulating, which is one main advantage to this class of drugs called DPP-4 inhibitors, which actually elevate levels and increase and promote their action. So this is one pathology which is addressed by those drugs. And whether so the neurotransmitters that are in the body can also regulate levels of these hormones and the secretion of these hormones. However, not in a very clear way, I would say. So the secretion of these hormones in response to neurotransmitters is still being resolved. So I would say in order to address whether their stress is related to their secretion and so forth, I think this is still being investigated. So
okay. All right. So I want to ask you about, another focus of your PhD was the drug Metformin, and how Metformin is one of the most common diabetes drugs. It has been around for years. One of the earliest drugs that were oral drugs that were available to treat diabetes. And the focus of your study was how it interacts with these peptide hormones, right? So can you tell us about that?
Yeah, sure. So Metformin, although it's been around for quite a long time and has been shown to be relatively safe in people, its mechanism of action is still not really well understood. So to date, most people still believe, and I believe also is the case, that Metformin acts in the liver to control the amount of glucose that is being pumped into the circulation. So when you reduce this, so Metformin acts by reducing this action of the liver, and then this effectively lowers the amount of glucose in the blood. So this is in general beneficial for type 2 diabetes. But one interesting finding that came out when I was doing my studies was that Metformin, when it's taken by healthy individuals, that it actually elevated levels of this in the circulation of this hormone, which I was interested in at the time, which was GLP-1, one of the incretin hormones. And so our big question was, since the mechanism of action of Metformin is not so well understood, is it acting through this other hormone, which is also beneficial for diabetes? So we chose to address this. This was part of my PhD work. And we found that, in fact, GLP-1 and Metformin have some overlapping functions. So not only do both lower blood glucose, but both also cause a reduction in the amount of food that one desires. So food intake is reduced when one is exposed to either of those two drugs. So then we asked whether, if you give Metformin to animals, whether they, first of all, whether we could see whether GLP-1 goes up, and we could. And next we asked whether the ability of Metformin to reduce food intake requires GLP-1. And in this case, we couldn't see that that Metformin requires GLP-1 for its action in that case. And then next we asked whether Metformin requires GLP-1 to reduce blood sugar. And we were also disappointed to find that although there was a strong elevation of GLP-1 when you give Metformin, there was no difference whether the mice had the ability to react to GLP-1 or not. So although GLP-1 goes up strongly when Metformin is taken, not only in animals but also in people, the action, at least according to our studies, doesn't seem to require GLP-1.
Okay, all right. So that's really interesting. We will move on to some of your more recent work. Yeah. So you have been recently more involved in studies that are looking into the intake of dietary protein and the different quality and quantity, and how protein intake sort of affects the metabolic health. Right? So this is a super interesting topic. We would definitely want to know more about that.
Yeah, so when I started my work in Heidelberg, I was working with a scientist named Adam Rose in the lab of Stefan Herzig. And his main focus of interest was on how the quality and quantity of dietary protein intake influences metabolic health. And upon starting the study, I had this idea in my mind that high protein is good for metabolism and glucose control because it helps you to feel full. So it promotes satiety, we say. And so my guess was that if we designed some experiments to look at different amounts of protein in the diet, that this would be the case where animals would perform best in metabolic challenges. So the main question was to address not whether eating less calories, not whether less calories was beneficial, but if we could influence the amount of protein intake by diluting it with other calories. So in this case, we were looking at carbohydrates. So we had groups which got very low protein and high carbs, groups which got average amounts of protein and average amount of carbs, and then groups which got high amounts of protein and low carbs. So different dilutions of protein. And what we found, interestingly, was that while the group which received the high amount of protein was eating less, which is what we had guessed and what we see often in people, the drawback for that group was that these animals were actually gaining more weight and that they were not performing so well in metabolic tests, like when we challenge with glucose, for example. So normally when you ingest glucose, your body clears it very quickly in a healthy situation. But when in animals that were being fed higher amounts of or more concentrated protein in the diet, they were performing poorly. So their glucose was actually going up higher. And conversely, when we looked at groups which got less protein in their diet and more carbohydrates, we saw that they had actually higher levels of energy expenditure, so they were burning more energy. They lost some weight as a result, although they were actually eating more. So this was one very interesting finding. And the other interesting finding was that these animals were very, very sensitive to insulin, and they had very, very good glucose control. So it just was a contradictory finding to what we had expected. And so we spent most of the time after that key experiment to investigate why is this happening, especially in the low protein or protein diluted diet. Why is it that animals are performing better? And we spent more time to investigate why this was happening.
All right. So to sum that up, you had two groups of animal groups, one on a low protein, high carbohydrate diet, and then the other one is in a high protein, low carbohydrate diet. So your finding of your study was the animal group with the low protein and high carbohydrate diet were doing better in the metabolic profile. They could, in response to glucose intake, their body could respond more quickly and better, and also in other areas they did better performance. So this is a very interesting finding of your study, because we see lots of different diet directions where there is a high protein diet intake is supposed to be good also in weight loss, but your finding was your animals, they were gaining more weight, right? So this is super interesting. We want to, what's your overall take on this? Obviously you told me before that it's a study. Every study has its limitations, and it was also not a study with like thousands of participants or any such thing. But tell us what's the implications also for humans from this study.
So at the end of after we did some investigations and tried to understand the mechanism of how this works, we found that one key hormone which was involved, aside from insulin, was this hormone produced by the liver. And basically when animals eat a low protein diet, this hormone goes up in the blood, and this plays a really strong role to improve their glucose control and to elevate their energy burning capacity, and causes them to lose weight. So we decided, okay, well, is this a phenomenon that's restricted only to mice, or is this something that's also relevant for people? Because ultimately we want to find new therapies and mechanisms to even dietary programs to treat type 2 diabetes in people. So we conducted a small study with collaborators in Denmark, in fact, where they took 5 healthy men and exposed them just to a standard protein diet for a couple of days, which is about 20% of their daily caloric intake, and measured some basic metabolic parameters like glucose and insulin in the blood and their body weight and how much food they took in and so forth. And then switched them to a lower protein diet, and then after 5 days, reassess them. And what we saw very interestingly was that even in people in this short study with 5 participants, but had very pronounced response. So they had lower blood glucose in response to meal ingestion. They had higher levels of this hormone I spoke about, which is FGF21. And what we could observe as well was that these people could, these improvements in their metabolism were reversed. So when we switched them back to their standard amount of protein. So what we could gather from this study was that the metabolic improvements that we could observe, at least some of them, which we could test in this short period, in terms of glucose control, were consistent in humans, and that one would need to maintain this program, this dietary regime, in order to keep the benefits. Because, like I said, when we switched them back, most of the metabolic improvements were disappeared. So, in general, I would say that diluting the amount of protein in the diet is probably beneficial, at least in this context. However, definitely more studies need to be done in other populations, for example, in people with diabetes, for instance, because in this case we were dealing with healthy people, and other populations like the elderly, and to see really if this is a safe approach for dealing with diabetes, and as well whether in the long run people respond well to it. So. Yeah, so more work needs to be done in this regard to better understand.
Also, the long-term effect has not been extensively studied, right?
Exactly.
What happens,
So we have a lot of work to do in that regard to better understand this regime.
All right. So another thing you have mentioned is the quality of the protein, the protein profile was also interesting in this study. I would really like to know a bit more about this.
So, when you digest proteins, you intake them as, let's say, long molecules, and then the body breaks them down with enzymes into single units, which are amino acids. And in the body, or in nature, there are generally 20 principal amino acids, and these are categorized into 2 types. So the essential ones, which you must intake in your diet because your body cannot produce them, and the non-essential ones, which you also intake through the diet, but the body can also produce them when you don't have enough intake. So these 2 classes. So in our studies that we had conducted, we generally just took the amount of dietary protein and reduced all the amino acids, and that's how we saw these metabolic benefits. So we reduced all of them. But of course, the question was whether it was the reducing specific ones or specific classes of these amino acids, which is triggering these metabolic effects, or whether it was
all of them.
Yeah, all of them. Whether you needed all of them or some of them. So we tried to address this, and going back to your point about amino acids in terms of meat-derived protein and amino acids coming from meat, I would say they're generally richer in these essential amino acids,
which your body cannot produce.
So generally, people who don't consume meat have to be more careful to eat specific vegetarian products to make sure that they're getting their essential requirements, let's say. So what we tried to do initially was to address whether a specific essential amino acids are involved. So what we did was we took diets that were low in protein, like we did before, and we gave back specific essential amino acids. And we saw that some of the improvements in metabolic control were reversed. So namely, in body weight changes. So we could see that the animals actually started to gain more weight when we gave some essential amino acids back. But other improvements in terms of glucose control were not changed. So we still have more work to do in that regard to understand how low protein intake or protein dilution in the diet improves glucose control.
Okay. How about in the context of diabetes? People with diabetes, what would happen if they would intake really low amounts of protein? Is that good or not good for them?
So at this stage, it's hard to clearly say. What we did observe, at least in data from rodents, with type 2 diabetes is that they have consistently higher levels of specific amino acids, so the essential amino acids. And this is also the case in patients with type 2 diabetes. So they have higher levels of specific essential amino acids. Now, whether this is contributing to their diabetes is a bigger question that we still need to address. So, of course, the issue that we need to address as well is that protein intake in the case of diabetes, in the case of elderly patients, is really important. So I would be cautious then to jump and say that all people with diabetes should reduce their protein intake, because there are some populations that actually need protein intake for their general health to maintain their, for example, bone structure and so forth. So I think what we need is perhaps more clear experiments to be done in people and perhaps specific populations to see which ones respond best in a safe manner to this kind of dietary change. So at the moment, I would say generally we need to do more to understand it better before we jump into prescribing this regime for everyone.
Right. There is no one size fits to all, right? It's also every individual is different, and also their dietary requirements, I would assume. What areas do you think there will be new things coming up?
Personalized medicine is becoming more pronounced as a strategy for treating diabetes, given that the population that has the disease is so big, and also to target those populations which don't respond well to the existing therapies. So a number of years ago, the lab started to do work to understand how the liver is contributing to diabetes. And it was found that one specific gene, known as TSC22D4, so it's a long gene name, is actually much higher in the liver in patients with type 2 diabetes. So some studies then were done to see whether if you target the lowering of this gene in the liver, whether this could be beneficial. And it was found in several animal studies that this was the case. So now the lab is trying to investigate how this is the case, or what is the mechanism behind this, to understand if this could be a novel therapy. As well, in addition to projects like this, the lab is also trying to understand, to identify additional molecules or genes in the liver which contribute to diabetes, and as not just in the liver but in other tissues. So we know that in diabetes is often associated with obesity and higher amounts of fat mass. So looking at other tissues like the fat mass to understand what's changing in that area in terms of regulation of genes and proteins is another area of focus. So aside from looking at these specific tissues, the lab is also interested in understanding how dietary changes can be beneficial. So for instance, one relatively hot fad at the moment is to change the timing in which people eat as a way of improving metabolic health. So this is often called interval fasting or time restricted feeding. So the lab is interested in understanding how, by restricting the time window in which you intake your food during the day, how this can influence your overall metabolic state. So in terms of your body weight, in terms of your ability to handle glucose challenges, ability to react to insulin, and so forth. So so understanding not only how this benefits the body, but also the mechanism behind it is a major approach, because to prescribe to someone that they should restrict their food intake to a specific time window is often, although we can explain the benefits to people, and I think people will be excited about it, the problem is often adherence. So people have a very hard time to stick to specific dietary changes for a long time. So that's why we're interested in understanding the mechanisms behind it, because in the case that the prescription of a specific dietary program doesn't work, at least we can say, okay, well, these mechanisms are changed in this dietary scheme. Perhaps we can somehow mimic those changes with other approaches, like with drugs or with yeah, that's how understanding the mechanisms is quite important, because we can come up with alternative ways to try to achieve the same benefits, but without the issue of people not sticking to the dietary program.
Yeah. That's always a big challenge. So talking about challenges, what do you think are the big challenges in coming up with new therapies for diabetes?
Because diabetes is so widespread, there's a lot of let's say drug classes that are emerging to treat the disease, or that have emerged. They all attempt to achieve the same thing, which is lowering of blood glucose, but in a safe way. Because as we know that if you have someone who in your family or a friend that is affected by the disease, you don't want to lower glucose too low to the point where it's dangerous and people can faint, for example. So achieving a safe level of glucose control is the aim. As well, because many drugs nowadays can do this, there are additional, let's say, requirements that drugs or therapies need to bring on board, which is including, for example, the lowering of lipids, which is also another problem in type 2 diabetes. So including cholesterol and triglycerides. So new drugs that are emerging not only are expected to achieve safe glucose control, but are also expected to reduce lipids. And as well, ideally should have positive effects on the cardiovascular system. So, for instance, another complication of diabetes is increased risk for stroke and cardiovascular disease. So there should be no risk that the drug will, or there should be no chance that a new drug will enhance the risk for these events, and in the best case should actually reduce the risk. So meeting all of these requirements, I would say, is a big challenge for people in the field of diabetes research or drug development, because although there are several candidates that are out there that achieve the glucose lowering, they don't, on the other hand, meet the other other requirements, which are safe for the cardiovascular system, safe for the liver, and so forth. So this is one of the major challenges, I would say, that's emerging. And as well, of course, they have to be superior in the best case to what's on the market, which has been, for instance, Metformin and the drugs which target the beta cells of the pancreas, which are already on the market and working. Whatever comes, any new players that come to the market have to be superior ideally in their glucose control, and have to be as safe or better than what's already there. And this is a big challenge for all companies and all researchers.
How about other areas such as stem cell therapy and other really new and emerging approaches? What's the prospect for these approaches in the field of diabetes?
With at least in the case of type 1 diabetes, stem cell therapy seems to be the most relevant, I would say, compared to type 2, in my opinion. However, as I explained at the beginning of our talk, type 1 diabetes is associated with lower levels of insulin and lower levels of the cells that produce insulin. The big challenge with simply replacing those beta cells, for example, with transplantable cells, for example, is that the body is actively trying to kill those cells in people with type 1 diabetes.
With an autoimmune reaction, right?
Because of this autoimmune issue that people have in type 1 diabetes. They can't simply be implanted with like an external pancreas or with stem cells that are differentiated into beta cells. So it's not so easy, although it would be nice. So the main issue is if patients are transplanted, then they need to be on immunosuppressant therapy for the rest of their lives, basically, in order for the beta cells not to be killed.
Survive,
exactly. And immunosuppression has a variety of complications associated with it, because it makes people more susceptible to other diseases. So in the case of type 1 diabetes, I think stem cell therapy, there are new things emerging to, let's say, shield beta cells or shield transplanted tissue from attack by the person's immune system. So these things, this technology is emerging, but one has to also always weigh in what is the benefit brought in by performing a surgery, which is invasive, implanting kind of foreign cells or tissue into the person, relative to their current lifestyle, which is in many cases involving several insulin injections per day and glucose checks per day, and so forth. So in all cases, the risk to benefit ratio has to be lower. So in order for us to proceed with that, and at the moment, I would say the majority of transplant cases, I would say, don't meet that requirement. So in most cases, I would say it's still leading to better quality of life by sticking with insulin injections and standard glucose measurements than going down the route of transplantation. In the case of type 2 diabetes, I would say, transplants are not as being actively investigated. There are other approaches people are using instead of transplants, rather to regenerate the cells of the body using peptide therapy. For example, this one that we spoke about earlier, which is GLP-1, or its analogs, or drugs that elevate the levels in the body by preventing them from being broken down. This is another approach to regenerate cells, let's say, by using drug molecules, which target the cells specifically, because in the case of type 2 diabetes, it's not autoimmune. So beta cells have the potential to be repopulated, let's say, to be expanded in type 2 diabetes. Of course, the major challenge is actually measuring this in people. So, for example, if we give a drug that we believe will enhance the number of beta cells in type 2 diabetes, in order to measure the beta cells in the person is a big challenge, because it involves taking biopsies or performing surgery to check this, and this is a major challenge at the moment, to assess whether they directly benefit the patient in that way. So again, we're trying to work on that to see if there are other, say, perhaps biomarkers that directly correlate
to beta cells.
So perhaps if circulating biomarkers which can correlate with the number of beta cells in the body, to avoid the need for biopsies and invasive measures to check this. So I would say that those are the major foci at the moment for looking at that.
All right. I often see patients who come with like sort of metabolic syndromes. In type 2 diabetes, a lot of them have the entire problems that you have already mentioned with triglycerides and cholesterol, and often with also problem with weight gain, which sort of correlates with type 2 diabetes. So what do you think about the approach because this is something we already know, that dietary and lifestyle changes such as exercise are very beneficial to type 2 diabetes. What does your research tell about this?
So in general, I would say, and I think many physicians would agree as well, that the first line of therapy, at least for when diabetes is first diagnosed, would be to change lifestyle by in promoting exercise and dietary changes, so reducing caloric intake. Whether lowering protein intake is also beneficial. This is something that we're interested to know, but hasn't been carefully looked at yet. The main issue is compliance in the end, because it's difficult for all people, I would say, to stick to specific changes to their diet or their lifestyle. So in many cases, the patients go home and come back after 1 year, recheck their glucose, and in the end, it's gotten worse or hasn't gotten better. So in that case, the physicians are required to take additional measures, which is involving usually metformin in the case of type 2 diabetes as a first therapy, or other drug options. So lifestyle changes, although are the cheapest way, and probably the best for the person's health, because they achieve very big effects in glucose control for people, and also have other benefits, not just with glucose, but lipids as well, and as well as mental health and well-being. So they promote exercise and dietary changes promote many benefits, which I think all people would agree is the best way to treat diabetes. Again, the issue is always compliance, which leads us to still need other therapies.
Yeah.
And our research is trying to understand, first of all, how these dietary or lifestyle changes, how they work in the body to promote metabolic health. In the case that compliance is an issue, that a patient cannot follow this dietary or lifestyle changes, how can we promote the same lifestyle, promote the same metabolic benefits, but through another means? For example, by giving a drug, for instance, to promote the same mechanism.
Yeah.
So that's generally what our research is focused on, I would say.
Okay. All right. That's amazing. So Adriano, do you want to add anything else to that? Because I think we have covered a lot of things. Yeah. Aside from, I would say, in general, leading the healthiest lifestyle one can lead is generally the best way for one to avoid the incidence of or being diagnosed with any kind of diabetes or type 2 diabetes in particular. I would say, leading a healthy lifestyle, exercise, balanced diet are the best ways. And of course, there are other methods which we are excited about to better understand, which involve time-restricted feeding or interval fasting or lower dietary protein dilution, which have been shown to have pretty interesting benefits. But all of these things still require time to better understand, and also to understand their safety in terms of in the long run. I would say those are the key points to leave with today. All right, thank you so much. This was very interesting. We got a very in-depth look at the mechanisms of diabetes, the different types of diabetes drugs, and I absolutely loved your knowledge, your in-depth knowledge on the topic. This was very cool. It was a pleasure to have you here.
It was my pleasure too. Thank you.
So this was Dr. Adriano Maida. We talked about diabetes and about his research work in the field of diabetes. If you find this topic interesting, I would highly recommend this episode that you watch it. And for those of you who have already watched, thank you for watching. And please leave a comment below and subscribe to the channel. I hope to see you in the next episode of Ask a Neurologist with a new guest. Thank you.